Hey guys, welcome back to my channel One Stop Biology. So today we are going to start with Kingdom Monera. Now Monera is a kingdom of prokaryotes. Okay, so this being the earliest form of life, the monerans are basically adapted to all types of habitat. We are going to discuss each and every detail of Kingdom Monera. So we'll start with the classification. Basically, there are two major groups of monerans. One is eubacteria, which is also known as true bacteria, and archaebacteria, which is ancient bacteria. And then eubacteria again is divided into two different types, one being bacteria and the other being cyanobacteria. So in this video today we are going to discuss bacteria. Now what are bacteria? Okay, so bacteria basically are group of prokaryotic organisms. So you'll have to remember certain terms like it is group of prokaryotic organisms. Now they are characterized by peptidoglycan wall. They have naked DNA and reserve food made of glycogen and fat. So there are three important parts of this definition. Basically, what are bacteria? They are prokaryotic organism and they are characterized by peptidoglycan wall, naked DNA, and reserve food made of glycogen and fat. So basically, it was discovered by Leeuwenhoek in 1676. Okay, so let's start with different features of bacteria. So they are unicellular. Okay, they have peptidoglycan cell wall. Then they have mucilage covering. Then they have circular DNA, which is naked and it is folded to form nucleoid. Okay, then let's move to even further features. Some being they have sap vacuoles absent. Instead of that, what they have is gas vacuoles. Okay, so they don't have vacuoles, they have gas vacuoles. They have membrane covered cell organelles like endoplasmic reticulum are absent. Okay, then they have 70s ribosomes, and for them the mode of multiplication is binary fission. So these are certain features of bacteria. Now they also have variety of nutrition, so they have varied nutrition. Now that includes they can be photoautotrophic, they can be saprophytic, they can be parasitic, they can be chemoautotrophic. So basically they can synthesize nutrition from sun, they can be parasite on some other organism, they can live on decayed material, they can synthesize food through chemicals. Okay, now let's move on to occurrence. So where do these bacteria actually occur? Okay, so they are most abundant organisms. Okay, they are the most abundant organisms. That means even a handful of soil contains hundreds and thousands of bacteria. Then they are ubiquitous. So they are found in all places where organic matter is present. They are found in all places where organic matter is present. So it can be in water, in soil, in air, maybe outside or inside of various, like bodies of various organisms. So they are present everywhere. Then they can tolerate extreme environment. They can survive in extreme environment, like they are present in hot springs. So they are basically present in very hot temperatures. Then they are present in frozen waters. So they can even survive in extreme cold environment. They are present in deserts which is deprived of water. They are present in oceans, deep inside the oceans. They can survive in acidic, alkaline, and saltous conditions. So basically, they are the most abundant organisms. They are present everywhere, and they can tolerate extreme environments. Okay, so this ends the occurrence of bacteria. Now let's go towards the structure of bacterial cell. So have you thought about the structure of a bacterial cell? What is the exact structure? Can we see a bacteria through a naked eye, or if the size is very small? So I'll explain you the structure of the bacterial cell. So basically, they cannot be seen from a naked eye. They are microorganisms, and the average size of a bacteria is somewhere around 2 to 6 micrometer in length and 1.1 to 1.5 micrometer in width. Okay, so this you will have to learn these things. So the average size of a bacterial cell is 2 to 2.6 micrometer in length and 1.1 to 1.5 micrometer in width. Then let's take some examples, like the smallest bacterial cell can even be of 100 to 200 nanometer. That means zero point 0.1 to zero point 0.2 micrometers. Now the smallest bacteria which is, so Dialister pneumocentus, this is the smallest bacteria. It has a size of 0.15 micrometer. And then we have the largest, so this is the smallest. Okay, then we have the largest bacteria. So largest one is spirochetes. Okay, so spirochetes, and it can even be blue green algae, wherein the size can reach up to 500 micrometers. Okay, so now you can see the diversity in size as well, right? So for example, there are certain bacteriums which are even 750 micrometer long, like we have Thiomargarita. So Thiomargarita is a virus which is 750 micrometer. Okay, so now we'll study is shape of a bacteria. So in shape, again, bacterial cells are of many types. Okay, so we'll take it up one by one. Now, so we'll start with coccus. Now this coccus means berry shaped. That means spherical or ovoid. Okay, so coccus bacteria are spherical or ovoid in outline, and depending upon their grouping, how they are grouped, they can be monococcus, They can be Diplococcus, they can be Tetracoccus, Streptococcus, Staphylococcus, and Sarcina. So basically, monococcus is occurring single, Diplococcus is two cells, Tetra is four, then Streptococcus means in chains. So you can see the form is in chains. Now, Staphylococcus basically is irregular clusters. So this is single, this is double, this is four, this is basically in chains, this is irregular clusters, and Sarcina is wherein they are present in 3D structure. Okay, so this is all about coccus, round, spherical, or ovoid bacteria. Then we have bacillus. Now, bacillus basically is rod shaped. Okay, so here the bacterium is straight and cylindrical like a rod. So it is straight and cylindrical like a rod with ends being flat, rounded, or cigar shaped. Okay, so they are cylindric, straight and cylindrical like a rod, and the ends being flat, rounded, or cigar shaped. So basically, it has three special types. One, it's diplobacillus. Okay, so wherein they are present in two. Then we have palisade bacillus. So this structure here is palisade. Okay, now palisade means like a stack. So they are stacked one at one another. Then we have streptobacillus again in chains. So this is streptobacillus. It means it is in chains. Okay, then the third form is spirulum, means in coil. Now the bacterium here is coiled like a corkscrew, and example of it is spirulum. Now here there's no types, it occurs individually. Okay, then the next form that we have is vibrio. Okay, so vibrio basically is like a comma shaped, right? So it's like a comma shaped or a curved rod. Now here the example is vibrio cholerae. So basically the cholera virus is vibrio shaped. Okay, now again like spirulum bacteria, here the vibrio form is live singly. Okay, then we have stalked, we have budded, and mycelial. So here the shape is of a mycelium. Here it's stalked, so it possesses a stalk. Here the bacterium is basically swollen at places. Okay, so all these forms like spirulum, vibrio, mycelial, stalked, or budded, they always live singly, whereas coccus and bacillus are two forms wherein we find different varieties even in the shapes. So they can be mono, diplo, tetra depending upon the number of cells joined together. Okay, so this is all about the shape of the bacteria, and lastly we'll cover the flagellation as well. So basically, depending upon the presence or absence of flagella, again bacteria are grouped into flagellate or non-flagellate. So they either have flagella or they don't have flagella. Now flagella basically is required for movement, but it doesn't mean that if the bacteria is non-flagellate bacteria, it will not be able to move. It will then to move, but flagella helps in movement. Okay, so there are various forms of flagellation again. So let's see the different forms of flagellation now. So these are the different forms of flagellation. Now here we'll start with atricus, which is here. This is the atricus form, this one, okay, which means flagella is absent here, okay. So this basically is atricus. Then we have monotricus. So here, as you can see in the diagram, there is one flagella present which occurs at or near one end of bacterium. So either of the end there will be one flagella, and it is known as monotricus. Second form that we have is amphitricus. Now what exactly this amphitricus is? So here the flagellum is present at each of the two ends. So there's one flagella and it is present at both the ends. So in all together there are two flagella, one at both the ends. Then we have Lophotricus. Okay, so Lophotricus basically means a group or tuft of flagella which is found only at one end. So here now there are certain minute differences here, like monotricus and amphitricus differ here. As in monotricus there is one flagella present at either of the end, and amphitricus there is one flagella present at both the ends. Then again Lophotricus has a group or flagella present at one end, and Cephalotricus has group of flagella at both the ends. Okay, and then lastly we have Peritricus, wherein the number of flagella here are distributed all over the surface. So these are 6 different types of flagellation. So a bacteria can either be a non-flagellate, that is atricus, or it can be a different form of flagellate. Now it can be a monotricus, it can be a amphitricus, it can be a lophotricus, it can be a cephalotricus, or it can be a peritricus. So with this we end the basic features that a bacteria has, the properties, the shapes, flagellation that we have covered here. Now, so let's revise the entire topic once again. So we started with the kingdom Monera, that is, what is the kingdom Monera? It is basically a kingdom of prokaryotes, and there are two different types of monerans. One is eubacteria, also known as true bacteria, and the other one is archaebacteria, that is ancient bacteria. Now we'll cover all the types one by one, and then eubacteria again is divided into two, bacteria and cyanobacteria. Now we started with bacteria. So bacteria basically is a group of prokaryotes. Now just remember that they are prokaryotes. So prokaryotic organisms, and the basic features of bacteria are that they are group of prokaryotic organisms. What all things do they have? They have peptidoglycan wall, they have naked DNA, and they have reserved food made of glycogen and fat. Now it was basically discovered by Leeuwenhoek in 1676. So remember these dates as well. Then the basic features of bacteria is that they are unicellular, they have peptidoglycan cell wall, they have mucilage covering, then they have circular DNA which is naked, and this DNA then is folded to form nucleoid. Okay, then they have gas vacuoles instead of sap vacuoles. Then they have membrane covered cell organelles, but endoplasmic reticulum is absent. Okay, then the ribosomes are 70s. They basically multiply by binary fission. Then they have varied nutrition. So by that we mean that they can prepare their food or they can get the nutrition through various means, like they can be photoautotrophic, they can be saprotrophic, they can be parasitic, they can be chemoautotrophic, and then they have flagella, which is single stranded. Okay, then we studied occurrence. So they are basically most abundant microorganisms. Remember, this is very important. They are most abundant microorganisms. Then they are present everywhere, they are ubiquitous. So basically they are present everywhere if the organic matter is present. So they are present in water, in soil, in air, and they can tolerate extreme environments like hot springs, frozen waters, deserts, deep oceans, or any kind of alkaline, acidic, or saltish conditions. Then we studied the structure of a bacterial cell. Now, structure again, it is a microorganism, so you cannot see through them through a naked eye. The average size, remember this size, is 2 to 2.6 micrometer in length and 1.1 to 1.5 micrometer in width. Now the smallest bacterial cell can even be of 100 to 200 nanometer, that is zero point 0.1 to zero point 0.2 micrometers. And then we saw a lot of examples like Dalister is 0.15 micrometer, that is the smallest bacteria, and largest are spirochetes and blue green algae, which can be of 500 micrometers in size. Then we studied various shapes of bacteria, wherein we studied coccus and bacillus, where we have different forms again. So depending on the number of cells, then they can be like mono, diplo, tetra. Strepto means chains. Then irregular clusters like staphylococcus. Then there are 3D structures like sarcina. Again, in bacillus, they can be either in diplobacillus, so in twos, palisade, that is stack, and streptobacillus, that is in chains. Then we have singular forms like spirillum, which is coil, vibrio, which is comma shaped. Then presence of mycelium can be named as mycelial. If they possess stalk, they are stalked. If they are swollen at places, means there is a formation of a bud, they can be budded. And then at last we studied the flagellation, the presence of flagella. So they can either be a flagellate or non-flagellate. Non-flagellate is atrichous. So flagella is absent. Then monotrichous, wherein there's one flagella. Amphitrichous, wherein two flagellas there, both the ends, at both the ends. Then lophotrichous is a group of flagella or stack of flagella at one of the end. Then cephalotrichous is group of flagella at both the ends. And then last was peritrichous, wherein the flagella was distributed on the entire structure of the or the entire surface of the bacteria. So with this we end today's video. I hope you understood. This is a very basic topic wherein we start with bacteria's shape and structure, but you should remember it well, remember the size well. And if you have any doubt, you can definitely mention it in the comment and we'll take it up. And if you understood the video, please like it, share it with your friends, and don't forget to subscribe to my channel. Thank you so much.